Hi, today I am going to review the steps of the catabolism of foods, which is the breakdown of foods to provide energy. And after that, I'm going to focus on the catabolism specifically of glucose and the pathway that the catabolism of glucose takes, both in an environment that is readily oxygenated, which is what our bodies are under those circumstances most of the time. But there are some circumstances in which the oxygen supply gets a little bit depleted because of overuse of oxygen, maybe during some excessive work, possibly poor gas exchange in our lungs, our body not getting enough oxygen. So we'll talk a little bit about what happens under those circumstances and how our bodies can continue to get some energy if we are somewhat limited in oxygen. So the picture that you see here is a breakdown of how foods get broken down and enter the citric acid cycle. So I want to remind you that the majority of our energy comes from everything that happens below the formation of this acetyl-CoA. Our lipids get broken down into fatty acids and glycerol. The fatty acids become oxidated to form acetyl-CoA. The proteins break down into amino acids and they undergo a series of steps that turn into acetyl-CoA. And the carbohydrates break down into glucose and other sugars and undergo a process called glycolysis, of which forms acetyl-CoA. This acetyl-CoA enters the citric acid cycle and goes through 8 steps. And at the end of the 8 steps, a little bit of energy has been released. Not much, just a little. I think it's only 1 or 2 molecules, maybe 3 molecules per glucose. It's pretty small. Also, some carbon dioxide and water is released. This process requires oxygen in and outputs the carbon dioxide. That's where this carbon dioxide is coming from. But the most important aspect of the citric acid cycle is that it spits out our high energy carriers, which are our reduced coenzymes, which include NADH and FADH2. All right, so these reduced coenzymes become oxidized and the initiation of our 4th stage, which is ATP production, gets started through the electron transport chain. So this occurs on the inner membrane. Whoops. It occurs on the inner membrane of the mitochondria. And there are some embedded proteins in there. This is just not designed to be accurate, just a visual representation of what's happening. And if this is the outer membrane of the mitochondria, this is called the inner membrane space. So it's between the inner membrane and the outer membrane. So what happens is these become oxidized and give off electrons, and I will represent the electrons as little dots. These electrons start to bounce from protein to protein. That provides energy to push out the I don't know what's happening. Zoomed all of a sudden. It produces energy which pushes out hydrogen ions. This is not going to work. I'm just going to redo my electron transport chain up here really quick. So I have the inner membrane here. I have the outer membrane here. And in the inner membrane, we have some proteins and also a channel. And what's happening is our reduced enzymes, NADH, I'll just use that one for now, is giving off electrons and hydrogen. So it's actually turning into NAD plus. And as the electrons bounce from protein to protein in the electron transport chain, hydrogen ions get spit out, and they start to build up in this outer membrane space. This channel right here is called ATP synthase. And when the concentration of these hydrogen ions builds up in this inner membrane space, the hydrogens start moving through this ATP synthase channel and provide the energy to turn ADP into ATP. So the hydrogen ions kind of make their way through kind of like a conveyor belt or a paddle wheel. And as they are going through, it provides the energy. That flow of hydrogen ions provides the energy to add a phosphate group to adenosine diphosphate. And it adds the phosphate group to the adenosine diphosphate to produce the adenosine triphosphate, which is a high energy molecule. So in this electron transport chain, our ultimate outcome is ATP. Now this ADP to ATP process is called oxidative phosphorylation. So that was citric acid cycle and oxidative phosphorylation in the electron transport chain in a nutshell. Again, the catabolism of all foods ultimately lead to the production of this acetyl-CoA, and therefore I probably will not talk about this process in much detail anymore, but we're going to talk about what happens with carbohydrates and proteins and lipids. So today the focus is carbohydrates. Before I go to the next screen, I do want to mention that the first step after carbohydrate digestion, in which we have the glucose and the other simple sugars, is a process called glycolysis. And through glycolysis, glucose is turned into pyruvate. And that pyruvate then gets transported into the mitochondria because this glycolysis is happening outside the mitochondria in the cell's cytoplasm. I do not know why random lines are showing up on my my thing here. Another thing about glycolysis is that this is an anaerobic process, which means it does not need oxygen. All right, so glycolysis occurs outside the mitochondria in the cytoplasm. It is anaerobic, which means it doesn't require oxygen, and it is also called an Embden-Meyerhof pathway. I don't know if I spelled that right. I know I spelled it right on the next slide. Okay, so talking about carbohydrates, the very first step in carbohydrate catabolism. Now, this is kind of going to talk about how glucose is broken down and also how it can be created. So, in addition to talking about the breakdown of glucose in a way that it turns into acetyl-CoA, enters the citric acid cycle, begins electron transport, ultimately ending up in oxidative phosphorylation to produce ATP. We're also going to talk about what can happen to recreate glucose, to put glucose back together. So glucose initially in the glycolysis process gets turned, before glycolysis, it turns into this glucose 6-phosphate, and that is essentially nothing you have to remember. So we have glucose through the act of glycolysis turning into pyruvate. So glycolysis is the biochemical pathway that breaks down a molecule of glucose into 2 molecules of pyruvate, plus there is a little bit of energy given off. Not much, just a little. Okay. Glycolysis is an anaerobic process. It does not require oxygen. Once the pyruvate forms in the cytoplasm, it actually makes its way into the mitochondria, and at the mitochondria it gets turned into acetyl-CoA. This S again is related to a sulfur atom that connects the acetyl group, which contains 2 carbons, to the coenzyme A. So the sulfur is just a connector in between the two. That's why sometimes we see it and sometimes we don't. The acetyl-CoA is then entered into the citric acid cycle, after which our high energy reduced enzymes, RE, electron transport, and we get oxidative phosphorylation that turns ADP into ATP. Okay, so this process, actually everything beyond the production of pyruvate, requires oxygen. It requires oxygen. So this process as a whole is referred to, I do not know what's happening. No, I don't know. It's telling me that I want to end my stuff. It's driving me crazy. I don't know what's happening with my pen. Anyway, this process of producing ATP, oh, look at how exciting that is referred to as aerobic cellular respiration. And it can be coming from glucose, as we're talking about here, or it can be coming from the lipids or the proteins. The process of ultimately producing acetyl-CoA, citric acid cycle, and electron transport time is aerobic cellular respiration. All right, so I am going to go back a little bit because I do want to talk a little bit about this step here. So once your body breaks the carbohydrates down into glucose, it can do one of two things. So it has two options. And the same is true for fats and also proteins. It can be used to build larger molecules, or it can be used to make energy. So in this option here, the glucose doesn't necessarily need to follow the energy pathway to produce ATP. It can also be put back together in the liver to be stored as glycogen. And remember, glycogen is the highly branched source of glucose that our bodies go to when the glucose levels in our blood get low. All right, I think that I need to, oh good, I thought I was gonna have to erase all my ink. All right, now there is another situation in which, let's say for some reason, we don't have enough oxygen to turn the pyruvate into acetyl-CoA. And what happens in that case, and it could be, again, maybe you are working really, really hard and your muscles are using up the oxygen and that oxygen starts to become in short supply. Or maybe the air exchange system in your lungs is not effectively taking in enough oxygen or releasing enough carbon dioxide. So for whatever reason, we don't have enough oxygen to continue. What can happen under anaerobic conditions, which is low to no oxygen, is our pyruvate can turn into lactate. Now, this process produces a small amount of ATP because glycolysis produces a small amount of ATP. So even if we don't have a lot of oxygen, our bodies still have a mechanism to make ATP through glycolysis. So what can happen is the glycolysis can occur, the pyruvate turns into lactate. So in the absence of oxygen, we can get a buildup of lactate in our muscles and in our body systems. And it can result in lactic acid acidosis, which can cause some muscle cramps. So if you've ever been working out really hard, you might get some muscle cramps. You are probably building up the lactic acid in your muscles. Now, when that oxygen supply becomes replenished, the lactic acid can turn back into pyruvate, and we can turn back into pyruvate, and we can actually do one of two things at this point. That pyruvate can turn back into glucose. And this is called gluconeogenesis. Neo is new. Genesis is to create. Think of, with biblical references, Genesis is the first chapter in the Bible, and it talks about the creation of life. So glucose, sugar, the creation of new sugar. So pyruvate can be turned back into glucose, and sometimes we don't need as much energy in our bodies. So We don't need the glucose to enter the ATP cycle. So glucose gives us energy. If we are limiting our intake of carbohydrates, we can turn pyruvate back into glucose. Now, one thing that we're going to learn is that amino acids can make pyruvate, and so can the glycerol part of the fatty acid can also make pyruvate. So we can get the new creation of glucose from non-carbohydrate sources. So our lipids can produce carbohydrates with the glycerol, and so can our amino acids from proteins. Now, if we are going to store our glucose, we need to turn it into glycogen, and that is called glycogenesis. So glyco refers to glycogen, genesis is to make. So the storage of glucose is called glycogenesis. So we make glucose into glycogenesis, or into glycogen. It's called glycogenesis, the creation of new glycogen. Now, if we need to go to that glycogen that is stored in our liver and our muscles, because the blood glucose levels in our blood are getting a little bit too low, so we need to replenish some of that glucose, we go to our muscles and we go to our liver and we break that glycogen down back into glucose. And that's called glycogenolysis. Glycogenolysis. Lysis is to break. Genesis is to make. And glycogen or lysis is to break. So our bodies have a natural regulation of glucose through two hormones that we're going to talk about, including insulin and glucagon. So our normal fasting blood level glucoses range from 60 to 110 or 70 to 100. It depends on the source. You're going to see different numbers for this. If our blood level glucose levels get too high, it's called hyperglycemia. And what happens is we get an increase in blood glucose. So we have too much glucose in our blood, too much. See what this is? This is lots of glucose. It reduces insulin, and the insulin causes the glucose to enter cells faster. If we're entering cells, we're removing it from the blood. It speeds up glycolysis, or the turning of the glucose into pyruvate. Pyruvate. Or it takes the glucose out of the blood and starts to store it as glycogen in our liver and skeletal muscles. So we've got three mechanisms, all triggered by the release of insulin. Our cells start using up that glucose faster. That removes it from the blood, so it enters the cells. Glycolysis speeds up, which is this process right here. So we get the glycolysis going into pyruvate, which can turn into the citric acid cycle. So the energy gets used. And also, we can increase the synthesis of lipids and proteins. So this glucose can turn into lipids and proteins. So insulin lowers glucose levels in our blood by increasing glycolysis, having cells take the glucose in. turning that glucose into glycogen or turning that glucose into lipids and proteins. Now, when our blood levels get too low, all right, and I'm just going to have like 2 glucoses there, so not a lot. We get a decrease in our blood glucose. It is called hypoglycemia, and it releases a hormone called glucagon. What glucagon does is it kind of stops or eliminates the glucose from entering cells, so it slows it down. Our glycogen starts to break down, which can free up some glucose. So the glucose from the glycogen is coming. So the breakdown of glycogen, the catabolism. And we can also get some gluconeogenesis coming from lipids and proteins stored in the body, which are also introducing some more glucose into our bloodstream. Gluconeogenesis, which is the production of glucose from non-carbohydrate sources. So this is just in summary how our body regulates glucose. This slide here summarizes all of it. And if I were going to offer any advice, it might be to copy this slide down, because it really does include all of it. So starting with carbohydrates. Carbohydrates break down into glucose. That glucose breaks down into pyruvate. A little bit of energy is produced. And this process, glycolysis, which is what this is, glycolysis occurs under anaerobic conditions. And it's also called the Embden-Meyerhof pathway. If there is not enough oxygen, we have anaerobic conditions, we produce lactate. So there's not enough oxygen to continue the Krebs cycle. This can result in lactic acid buildup and some acidosis or muscle cramping. If that oxygen returns, we can turn that lactic acid or lactate back into pyruvate. At this point, the pyruvate can enter in an oxygenated environment into the production of acetyl-CoA and begin the citric acid cycle, in which we get a little bit of energy. We get some carbon dioxide. Looks like I'm missing something there. We produce these reduced enzymes, which make their way down the electron transport chain to give us oxidative phosphorylation and ATP. If our body doesn't have enough glucose, what can happen is our we can get what is called gluconeogenesis, where we take pyruvate or other non-carbohydrate sources and we create glucose. So gluconeogenesis is the pathway for synthesis of glucose from non-carbohydrate sources. And the question is, what if we don't eat enough carbs? Our body makes glucose from the fats and the proteins that it needs. It can also get glucose from our glycogen stores. So we can get the production of more glucose either by breaking down the glycogen or gluconeogenesis. If we have a little bit too much glucose, our glucose can get turned into glycogen storing. Another thing that can happen is if we can't make any more glucose, the glucose can get converted into lipids and be stored as fats. So if we are we're eating a lot of glucose and a lot of fats, a lot of calories, and we're sedentary, so we don't need as much adenosine triphosphate. We don't need as much energy as what we're eating. Our body turns it to storage. And that storage includes glucose going to glycogen and ultimately glucose turning into lipids as adipose tissue. So it makes you gain weight and store energy in the form of fat. The definition for glycogenesis, the synthesis of glycogen in the liver, excess glucose can lead to this. This is that glycogenesis formation. And this is just a couple of notes here. Glucose is the most important monosaccharide for ATP production. And the aerobic oxidation of glucose supplies more than 90% of ATP for our cells. Glucose is the primary energy source for our cells. Glucose is the preferred energy fuel for our brains. Our brains can run on glucose, and it can also run on ketones, which we will talk a little bit about when we refer to fatty acids or metabolism, the breakdown of fats. So I encourage you to copy this down. It's an excellent review of just about everything you need to know about carbohydrate catabolism and anabolism. So we have carbohydrate anabolism here, the building up of glucose into glycogen, of glucose turning into lipids. So glucose can get turned into bigger molecules, and it can also get broken down and enter the citric acid cycle. So I think that is my finale on glucose catabolism and anabolism. Thanks for watching.